within the plant body, histologically, the tissues are meristems and permanent tissues. These tissues have got organized in a characteristic manner within the plant, and when we study the anatomy of the stem, the root and the leaf, we find that these tissues have assembled in a characteristic manner, and based on their assemblage, their function, they are broadly grouped into three categories. The three categories of these tissue systems in the plants are epidermal tissue systems, ground tissue systems, and vascular tissue systems. Epidermal tissue system, as the name signifies, epidermal refers to the outermost covering of the whole plant body, and this epidermal tissue system consists of epidermal cells. Just as the human body has the skin, plants also have an outermost layer covering their entire body. Though the epidermal cells are parenchymatous, they have got a wide range of activities, as the epidermal cells sometimes modify into the guard cells guarding the stomata or the openings. They do have lot of appendages in the form of multicellular hairs called trichomes, or unicellular hairs like the root hairs. The epidermis is the outermost layer of the primary plant body. It is single layered, parenchymatous, with small amount of cytoplasm lining the cell wall, and just like any other permanent cell has a large vacuole. In the leaves as well as very tender stems, there are present stomata, which are the small openings, and these stomata are guarded by modified epidermal cells called guard cells. The stomatal openings are regulated, and these stomata help in the process of transpiration as well as gaseous exchange. Each stomata in the case of the monocots has two buoyant-shaped guard cells which enclose the stomatal pore, whereas in the case of the grasses and most of the monocots, the guard cells are dumbbell-shaped. What is significant of these guard cells is that their outer walls, which are away from the stomatal pore, are thin, whereas their inner walls are highly thickened. The guard cells possess chloroplasts and regulate opening and closing of the stomata based on the amount of water that is present within the guard cells. When the guard cells are turgid and fully swollen, the stomata open, and when the guard cells are flaccid due to the loss of water, the guard cells close the stomata. That is how transpiration gets regulated in the plants. Sometimes a few epidermal cells in the vicinity of the guard cells become specialized in their shape and size, and these cells, which are the modified epidermal cells, are called subsidiary cells. The stomatal aperture, the guard cells, along with the subsidiary cells, together constitute stomatal apparatus. All the tissues except epidermis and vascular bundles constitute the ground tissue. In the leaves, the ground tissue consists of thin-walled chloroplast containing cells located between the upper and the lower epidermis, called the mesophyll cells. In the case of dicotyledonous leaves, the mesophyll cells that are close to the upper or dorsal epidermis are cylindrical, green, and are in one or two layers. They perform photosynthesis and they are called palisade parenchyma. Whereas those mesophylls which are present between the palisade parenchyma and the lower epidermis in the dicot leaves have plenty of intercellular spaces and constitute spongy parenchyma. In the case of the monocots, there is no differentiation of the cells in the mesophyll. They are all loosely arranged. They do have chloroplast, and all the cells perform photosynthesis. The ground tissue in the plant body comprises of parenchyma and those tissues which are derived from parenchyma. They fill the entire interior of the plant, forming cortex, the outermost layer of cells represented by parenchyma, sclerenchyma, as well as the innermost layer of the cortical cells called endodermis. The ground tissue also occupies the central portion of the stem, what is called pith, and in the pith are present loosely arranged parenchyma. The third kind of tissue system in the plant is vascular tissue. The vascular system, as already being explained, comprises of phloem and xylem. The xylem and the phloem together constitute the vascular bundle. In the case of plants, the vascular bundles are of different types. One of the most common types of vascular bundles is conjoined vascular bundle, where xylem and phloem are present together and they are in the same radius. Hence that vascular bundle is called conjoined. In a conjoined vascular bundle, it may be collateral, where the xylem and the phloem present in the same radius, having cambium in between them. Such a vascular bundle is called collateral conjoined open vascular bundle. This type of vascular bundle is present in the stem of dicot plants. The second category of collateral conjoined vascular bundle is the one found in monocot stems, where the xylem and phloem, they are present in the same radius, but in between them there is no cambium. Hence it is called collateral conjoined closed vascular bundle. The vascular bundles are also represented in a third form, what is called bicollateral vascular bundle. In a bicollateral vascular bundle, which is the characteristic feature of some of the plant families, like the family Cucurbitaceae, there is a central xylem which is surrounded by two patches of cambium, which in turn is surrounded by two patches of phloem. This is not a very common type of vasculature. The vasculature present in the roots is something entirely different from that of the stem. In the roots of both dicots and monocots, the xylem and phloem are not found together in the same radius. They are present in separate bundles, and they are in equal numbers, arranged alternately at different radii. Such a vasculature present in the root system of both dicots as well as monocots is said to be a radial arrangement or radial vasculature. Now, having seen these three different types of tissue systems, let us go to the details of how these tissues are assembled in the case of dicot and monocot plants.